Starting 2 minutes earlier today. Hello everyone, how are you today? Other than it feels a little chilly today. So today we are going to talk basically about the expression of the gene. So as we defined the gene as the area or segment of the DNA that can express the gene, they can make RNAs, right? That's the definition. Actually the correct definition of a gene. So then we obviously we have to look at what exactly then usually when we say the gene expression. It's a process of making RNAs, yeah, any type of RNA. As you have looked at it, there are six, well actually depending on how you look at it, there are even 7 different RNA existing these days. But anyway, all those RNAs making processes, we can legitimately say that's a gene expression. And all of those gene expression, by the way, that gene expression is called, that particular terminology we call the gene expression is transcription. Actually, the transcription is the process of synthesizing RNA from DNA using the DNA template. So in other words, the transcribed product, which is RNA, is actually always the complementary to the template DNA. Complementary, what I mean by that is, like, if the DNA reads like AGCT, then the RNA should read like U, and C, G, then A. Like those kind of complementarity you can always expect from any given RNA sequences. Obviously, any of those processes in cellular activity should be catalyzed by enzymes. So this time around, the name RNA polymerase is the one catalyzing the synthesis of RNA from the DNA template. Okay, so what else? So this is a cartoonic diagram of one of the simple version of the RNA polymerase in E. coli, the bacterial version of RNA polymerase. You don't have to really pay attention to any details of labeling, but anyway, just simply without giving you any detailed more additional information, if you take a look at this cartoon, then what you can probably understand is what the whole enzyme DNA polymerase is not a single protein but multi-subunit. So there is a one big omega, beta prime, and beta, alpha. Several different proteins get together to comprise one functional group. Many of those proteins work just like that. Most of the proteins simply don't work as a single product, but as a cooperative multi-subunit, so-called. And this is another such case. And this DNA, so as an RNA polymerase, it has to be able to, it has a capability of recognizing and grabbing this double-stranded portion of the DNA, right? They grab and open. Take a look at this. Take notice of this area. The DNA double helix area partially opened up. Excuse moi, and open up. And so this area opened by RNA polymerase temporarily became single-stranded now. And those each single-stranded, now look at what's happened. One area of a single-stranded DNA is now paired with RNA, right? So temporarily, DNA and RNA hybrid. Someone asked in the previous lecture question that is there any natural example that as a like existing as a DNA RNA hybrid? Yes, this is a perfectly good example of always it happens. This is DNA, and this red line represents the RNA. So between DNA and RNA, this complementary base pairing is being established. And as a result, this new RNA is being synthesized and released. So that's always a lot of, you will be surprised at how many different important cellular processes are actually based upon this simple complementary base pairing relationship. It's amazingly. That actual chemical nature of complementary base pairing is very straightforward and simple, but a lot of different cellular processes are simply based upon that and make all such a critically important process that sustain the life is being dependent upon such process. So anyway, that's how you do it. So RNA polymerase can recognize and grab DNA as well as also synthesize and then temporarily can hold some portion of the RNA, and that's their product. And obviously, what is this? NTP? N is here, the N is whatever. AGCT, 4 different, not AGCT, but in this way, the N should AGCU, okay? 4 different kind of RNA nucleotides. The NTP represents such a thing, like John Doe, everybody. So it can also recognize and can feed, provide raw material. If you want to synthesize the polymer, if you want to synthesize all this long polymer of RNA, then you need, obviously, this raw material, which is single nucleotides. So they are also being fed to the RNA polymerase. So actually, by just simply looking at this cartoon, you can say a lot about, you can offer a lot of different stories about this. That doesn't mean that I'm going to make a lot of those different stories in your upcoming exam. No, it's not. But anyway, that's just what I'm saying. So another, maybe a lot of you at this point then wonder, then who can become gene and who cannot? There are, along the long stretch of DNA, it's human DNA. You know how many different nucleotides are actually there in a human entire chromosome DNA? More than 3 billion pairs. More than 3 billion nucleotide pairs. Actually, if you try to put those 3 billion nucleotides as individual nucleotides as an alphabet and then try to like pretend as if that's just like alphabet, letter, books, then the page, the number of different pages of that such book will exceed it's a million page as a standard, the A12, A4, the paper size book. That much huge information is there, but unfortunately, not all those more than 3 billion nucleotides are actually participating as a part of the gene. So then, among this DNA area, what's the criteria of which segment, which region can act as a gene? On the other hand, the other huge area, like simply just sitting there and doing nothing, obsolete. As far as the gene perspective, they are not producing anything. So, and all of a sudden, another segment can act as a gene. So, who can become gene and who cannot become a gene? It's just a matter of whether you can have this or not. In the vicinity of any segment, if you think about it, DNAs are just a simple DNA or random arrangement of AGCT, or just different nucleotide. So randomly they are arranged. And then by even so, how can certain segment, which can act as a gene, another segment of DNA which cannot act as a gene? And what's the difference? Are there any differences in DNA the nucleotide arrangement? Not necessarily. As far as this RNA synthesis process is concerned, no. Simply whether you have this or not, promoter. Then what is the promoter? Promoter is landing site. The previous slide, which was about RNA polymerase. So RNA polymerase is an enzyme. So RNA polymerase need to be able to find, okay, that is the location that I want to start my job. So think about this in whole huge, in new nucleus. In a cell nucleus, nucleus is a three-dimensional space. DNAs are all arranged randomly. Okay? So DNAs are some, right? DNAs are some very steady line on the bottom, like a railroad-like structure. No, that is not. The DNAs are all like three-dimensional structure hanging around some different types of so spider web-like, rope-like structure. Maybe you can compare it. And among those whole different complexity of the DNAs in three-dimensional space, and this DNA polymerase should correctly be able to recognize the starting point where I can start this my job so that I can synthesize the RNA. Then there's got to be some kind of rule. Let's put it this way. Okay, promoter is some kind of starting point, starting line for RNA polymerase. Okay, that's my location. Whenever there is a promoter is what? Simply the sequence, specific arrangement of a specific DNA sequence. Okay? And that's promoter. That is specifically recognized by this protein RNA polymerase. Oh, I love that space, that sequence. So that this RNA polymerase can bind, recognize and bind, which we can easily compare to helicopter landing spot, something. Then now the difference can be made. Once this DNA polymerase land on there, and then this RNA polymerase land, then it can open up, unwind, and then start putting the complementary nucleotide on that template DNAs. That's how it starts and finish the process of RNA synthesis. So anyway, this promoter sequence is there. We can recognize specific promoter sequences. It's not really that difficult because these promoter sequences are fairly conserved among many different genes. So simply you sit there and then having a coffee, and by just simply looking at the computer window screen, and if you examine all DNA different sequences, and then, oh, it looks like this area, it looks like very close to promoter sequence. Anyway, that particular arrangement, specific combination of arrangement of the DNA sequence, although it's random but very specific, so it's not everywhere. And somehow throughout all the process of evolution, so that somehow became the sequence element that defined the start of the transcription. Once RNA polymerase bind, sit there, and then now start synthesis of RNA just right next to the promoter site. That's what it means. So promoter is located at the 5' proximal position. 5' proximal position? What do I mean by that? If let's say this arrow indicates the starting point of the transcription, and promoter is right at this point over here. So RNA polymerase will bind over here. Okay. So by convention, we usually arrange this DNA or RNA usually on the left side as this is a 5' end, and the right side is a 3' end. That's what it means. This promoter sequence is right at the 5' of the 5' upstream, proximal upstream of the starting point is what it is. Okay. So actually the importance of a promoter can be recognized by the presence of so-called pseudogenes. Pseudogenes are exactly the sequence itself. Internal content of this sequence is exactly, almost or exactly identical to known existing gene sequence. However, these pseudogenes can never be expressed. Why? Because it doesn't. It doesn't have any promoter. So there is no occasion for these pseudogenes to meet RNA polymerase to visit and then do this thing. That happens. Sometimes along your genome, actually, occasionally you can identify such pseudogenes. Yeah. How this pseudogen happen? People believe that actually it's just a remnant of some of the viruses. Virus-like DNA element, there is something. I don't want to go further. And through the activity, some segment of the DNA jump around, and then somehow lost their necessary element, which is a promoter. So now it became dead end over there. It cannot escape, and it just stuck there. Is the origin of pseudogenes. Yes. Exactly. Yes. 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 Some people actually play such a trick. It's called the enhancer trap or anything, or some different tricks. So you can try to insert randomly so the promoter sequences, and then somehow, wow, some genes can be expressed. Or the other way around also, people, in order to identify some such promoter element, also they do this trick. Yeah. Majority of the still don't understand what I'm talking about, so I will shut up this. So here, promoter. Just speaking of a promoter now, I can try to unfold a little bit more about this area. Overall, it's called the regulatory region of a gene. Okay. So let me read this. A typical gene consists of the transcribed area, one transcribed area, which is exactly identical in sequence to the synthesized RNA, right? So if this DNA, let's say this is a DNA, this area is DNA, and through transcription, an RNA was made. Okay. So this RNA was synthesized starting from over here. So exactly this RNA will be exactly identical to this area of the DNA. Then we can call this as a transcribed area. However, that is not the only part of the gene, because as we saw in the example of the pseudogene, just because you have only this the segment doesn't mean it will not be expressed. It cannot function as any gene. You need what? Additionally, promoter here. You have to have this promoter area. So this promoter area should be a part of a gene. However, can you see this promoter sequence in the produced RNA? No. It's just like, yeah, it never. It's simply behind there. Without this promoter, the gene is not going to be expressed, but express the gene itself, the gene product, the RNA, does not reflect the presence of this promoter, right? So, however, you cannot say this promoter is not part of a gene. Without it, you cannot. As a gene, you cannot function. So this should be definitely included as a part, important element of any functional gene, although it is not seen in the produced RNA. So then the question is, are there any other such things? Yes. Not only the promoter, but also you can expand this area usually further. So this is really a mysterious area, not really mysterious, but very interesting area, that there are other additional DNA elements which so to speak, absolutely necessary for the functional gene expression, in addition to the promoter sequence. So entirely, altogether, such this area, we can call this area as collectively a regulatory region of a gene. So entire gene can be expanded to to include such a regulatory area. Not only those what you actually see as a product of the RNA, which we call as a transcribed area, plus the regulatory area make up altogether the whole functional unit of a gene, is what I'm saying. So, and be more specific. Okay, you mentioned the promoter, then what else there in the regulatory area? Okay, then I will give you an example, for example, and such as enhancers or silencers are the very famous such regulatory element contained in the regulatory region. What? Enhancers and silencers? Yeah. At least they sound like, what is promoter? Okay, first we have to really make this clear. What is promoter? Promoter is what in chemical nature, is it what? DNA sequence, it's DNA. Promoter is some very specific order of a DNA sequence element. So promoter is a DNA sequence, and which is really needed for gene to function. And these two things are very similar. Enhancers or silencers are also the DNA sequences. Both of them influences the level of gene expression. So I will pair this enhancer and transcription activator, these two together. Because just like when I say promoter, then you automatically think of what as a partner? Promoter and what? RNA polymerase. Yeah? Promoter is very specific to who? RNA polymerase. Hemoglobin, who gives any about promoter? The protein hemoglobin doesn't care anything about the promoter. To me, it's nothing. Only to RNA polymerase, promoter sequence is very special because that RNA polymerase protein is the only one that can recognize special thing about promoter and then specifically interact, bind. It's always like that, life. In life's chemistry, life interactions are always based upon such specific specialty. You and I are very special. I can only recognize you, and you can only recognize me, and between you and me, we'll have a very, very specific relationship, usually culminating in binding. Okay. The special recognition always usually result in specific binding. Think of, I just mentioned this thing twice. I mentioned this earlier before, this same principle, which is very important and which is very widespread in all different life processes. This interaction, specific interaction through binding. And same thing, in this occasion also you are witnessing this promoter and RNA polymerase specific interaction. Now, enhancer, whatever it is another such specific DNA sequence. And this specific DNA sequence is recognized only by some specific protein we call as a transcription activator. Those are proteins, another protein. Those proteins are also additionally, the ones additionally needed for proper level of gene expression. Okay, so if you thought that RNA polymerase is the only one that is needed for such gene expression, then you are wrong, because RNA polymerase is just one, simply, yeah, absolutely required one, but with only this RNA polymerase, you cannot really, virtually, do any efficient or effective gene expression. You need something more. that is provided by a protein called the transcription activator. Or a lot of times this transcription activator is usually casually simply called the transcription factor too. Interchangeably, people use transcription factor or transcription activators, and those are the proteins that help. But in order to help for this transcription activator, help this gene expression, they also need to be able to sit on those gene area, just like RNA polymerase. They need some specific landing site. Then we call that specific landing site as an enhancer DNA element. So what they do, overall we can summarize their job as they facilitate transcription. Yeah, how? Well, some say, yeah, it can stabilize the RNA polymerase. It's a very dangerous job for RNA polymerase to hang on the rope-like DNA structure. Imagine that. It will be really, really risky, difficult job to do. You just hang on this DNA rope and then try to open these two double-stranded DNA, and then at one, if RNA polymerase has two arms, where one arm is grabbing this DNA, opening up, and another arm trying to put a new complementary nucleotide to each according to the sequence of template DNA, it will be really, really difficult, hard job. Yeah. So if there is some on their back, on the RNA polymerase back, if there are some reliable friends, like, oh, we'll support you, we will grab you, don't you worry, we are holding you, so that, not to fall down. Yeah, a lot of times actually RNA polymerase fall down in the process before they finish. Okay. Then they have to reclimb. Oh shoot, and they have to reclimb and then restart all these things. If that happens often, that means the efficiency of this RNA polymerase is very low, that means. So this gene expression is very low. But on the other hand, if this RNA polymerase doesn't really fall down, it can go continuously all the way down. And then we can say this RNA polymerase is doing a very good job. So, as a result, the gene expression level is very high. And that makes a difference. And what's the difference? I do have friends. The RNA polymerase who produce really high level of gene expression, what is the secret? We interview the RNA polymerase. What's the secret? Tell me. Then they said, they confessed. Oh, the secret is I have friends in my back. They are transcription factors. Yes. So that's the role of transcription factors that helps by stabilizing. Another very important job provided by the transcription factor is also this. This is far more important than this. Chromatin condensation is almost everything. If this if this chromatin exists as a heterochromatin, do you still remember the difference between hetero and euchromatin, right? Don't say no. Euchromatin and heterochromatin, what's the difference? Heterochromatins are the one highly condensed. Euchromatins are more distantly placed in the nucleosomes. And what's the big deal? What's the big deal between these two different status? These are open. These euchromatins are more open structure, so that more accessible the area for those enzymes like RNA polymerase. So those euchromatin area has a tendency of providing higher gene expression, obviously. Gene expression is highly correlated with the chromatin condensation, degree of chromatin condensation. If chromatins are highly condensed, gene expression would be very, very low. If chromatin condensation is very low, so that opened up, then gene expression, that area, the same area, that area's gene expression will go very high. And that differences, condensed or opened up, can they actually adjusted by the activity of these transcription factors? Then is really, really important function coming from this transcription factor. Transcription factors do that. So, in other words, practically speaking, without transcription factor, without transcription activator, virtually no gene expression can be made. You may still produce a couple of, several, 20 or 30 copies of RNA, but that's nothing from the cell scale. Are you kidding me? You only give me only 20 copies of this particular gene? Where am I going to use this? At least you should be able to provide me, if you ask me to express this, which means make the RNA until, at least you should provide me some several hundred or even 1,000 copies. That's sound exactly identical copies of RNA. That level we call as a okay, decent amount of gene expression. And in order to achieve such high gene expression level, you need the help of this transcription activator role. The promoter, RNA polymerase function alone cannot do that. So this overall, this area probably you can define as what? Such regulatory area of the gene. Let's say this is DNA, this DNA label. And so this area, on the other hand, probably the transcribed area. If you compare this transcribed area and the size of the RNA initially made should be identical, that matches. Okay. That's, by definition, you can call it as a transcribed area. And so, for now, simply only paying attention to this regulatory area. The promoter is there, which serving as a binding site for RNA polymerase. And now you also see. So another thing is upstream and downstream notation. What's upstream and what's downstream? So conventionally, we usually designate this arrow as a starting point of a transcription. So upstream means in relation to this starting point, the backside, we usually call it as upstream. Or to be more specific, we usually call this upstream as 5' upstream. And downstream is the far ahead side or downside of this starting point. And in order to, once again, to be more specific about this downstream, you put usually another additional 3' downstream, because by convention, we usually place any DNA segment as the 5 to 3' orientation. 5', 3', at this point, do you understand what's 5' and 3'? Yes? If not, ask me in your Chokji question, then I will repeat in question and answer section was 5' and 3'. But it's just a designation way of putting some relative orientation of any nucleic acid, whether it's DNA or RNA. So it's a conventional way of designating. So anyway, so 5' upstream, there are usually several. This is typically is a typical structure of a gene, eukaryotic gene. Prokaryotic gene structure is maybe at least with regard to this regulatory region, they are much simpler than those of eukaryotes. So we better actually see directly at first the structure of eukaryotic gene expression, so that because there is no such thing like the enhancers usually in prokaryotic gene. So in order to understand what's enhancer and what's transcription factor, probably it's better to directly handle this eukaryotic genes structure. That's why. So anyway, these areas are the regulatory elements, and those are specific proteins abound. So some of the transcription activators and RNA polymerase, RP, and there are more proximal, closely associated transcription factors. These are. So speaking of transcription activators, some friends, transcription activators, we can call them as a friends of RNA polymerase, right? They help, as a friends, they help the function of RNA polymerase in many ways. And some of those transcription activators work very closely associated with the RNA polymerase. And some others are surprisingly work such a far long distance ahead of them, far upstream, far from upstream, and still they make their, they do their job without any problem. And there is a negative element. So enhancer, if the enhancer, which is DNA sequence, is the one actually positively regulating the gene expression by recruiting those transcription activator protein. The silencer as a DNA element, once again, silencer is another DNA element, the sequence element, but it acts negatively in the regulation of the gene expression by recruiting at this point. The silencer is a binding site for what? Guess what? The negative the factor which we call as a repressor. Like there is an angel and a demon, yeah, and there is a transcription activator and the repressor protein act antagonistically always. This repressor protein will suppress, downregulate, whereas transcription activator will upregulate, increase. So they always will collide. Which will win is the outcome. Depending on who wins, the outcome of the gene expression will be determined, I guess. So if as a gene, if you want to actually achieve a high level of gene expression, first you have to get rid of this. Okay? Just like in your car, if you want to start your car, you have to take off your foot from the brake and then put the gas pedal. Then only then the car can go, just like that. So these two things have to be always taken care of in balanced fashion. Okay. So this diagram, this figure illustrates the function of such transcription activators. Here, notice that transcription activators are simply labeled as a transcription factors, as I said. This transcription activator and transcription factors are interchangeably used in many cases. So this is the transcription factor means transcription activators. Then what do you think it has done in this figure? Initially, the chromatin structure was like that of heterochromatin, highly condensed. And this area, at least this area, was opened up, exposed. This nucleosome, this individual things are individual nucleosome. These nucleosomes are tightly closed together, making the heterochromatin structure, and somehow these transcription factors somehow bound here and then expanded. Okay, move away. So this area became open so that now this promoter sequence was exposed thanks to that. Then RNA polymerase now can recognize this and then come over here and then do this thing. This is what has been illustrated. So these active gene and inactive genes, you can make distinction depending on the presence or absence of the function of these transcription factors. Okay? So active gene we can define as in this area the necessary transcription factors are available. They are bound to the promoter area. Inactive gene, why you're not doing anything? Okay, your gene, do your job. Remember how I defined the gene? A segment of a DNA that can make. It has a potential, but it doesn't necessarily have to provide always 24/7 like activity. If you want to idle, so be it. Then if you, as a gene, if you ask why you are not doing your job, because I don't have my necessary transcription factors. That's a very good excuse. Promoter is universal. I mean, RNA polymerase. RNA polymerase is universal, means for all the messenger RNA gene, same one kind of RNA polymerase takes care of all the messenger RNA transcription, just like that. However, each particular individual genes have a different taste for transcription factors. So that makes difference. Certain genes require some very specific set of transcription factors, whereas another gene, although both these genes are making proteins, mRNA gene, but this gene may require different kind of transcription factors. That's how it goes. And silence and repressors are just the combination of this partner that's simply doing the exactly opposite effects of the enhancers and transcription factors. So here, one student had the relevant question on this subject, asking that why the gene activity are not synchronized all together. Every possible genes are activated at the same time and silenced at the same time, because exactly, yeah, you're right. Along with your life cycle, lifespan, you only need certain particular proteins on a very given specific time or very specific different locations. We call it as such thing as a differential gene expression, and such differential gene expression is the standard norm for any multicellular organism. Even single-cellular organism, they do not need all their set of genes in a given time. Okay. They only need certain set of genes in a very specific given time. So you definitely need some kind of mechanism of only selectively activating some of the gene you need, silencing others that you don't need at this point. And somehow cells very cleverly coordinate such regulation. It's very amazing things. So this differential gene expression, then probably some of you have already answered. Okay, if that is the case, how can we? Remember, actually, as you're a creator God, how would you design this thing? So your cell, your creation, the cell, should be able to demonstrate this, the ability of differential gene expression. How? How would you implement this differential gene expression capability, given the information that probably you can utilize this? Remember, RNA polymerase is universal. If you try to simply modulate the activity of RNA polymerase, it will affect all or none, because RNA polymerase is universal. If you deactivate RNA polymerase, and then it will affect all the genes so that by this route you cannot achieve what you want. So then what's the choice? But luckily, what did I say about? The transcription factors are very specific. Transcription factors are specific to very different specific gene, so you can play this game using the transcription factors by making some very specific subset of the transcription factors activity only available to where you need, in space or time-wise, whatever, both ways. Then somehow you can achieve this so-called differential gene expression. So this, once again, in summary, differential gene expression can be achieved by this playing different type of transcription factors. So this particular transcription factors activity make them available only in where you need actually, or in particular time span you need, then you can achieve the so-called this differential gene expression. So what are the examples of so-called differential gene expression? One very straightforward example is something called a tissue-specific gene expression in multicellular organism. Like this, in different tissues. Why those different tissues look different? Because they function different, right? Of course. Or maybe the other way around. Because these different tissues look different, that's why they can provide different functions. But how? By, like for example, brain and your liver. Different gene products are made in your brain, whereas another different type of genes are made, expression is made in your liver, then the function of the liver will be quite different from those of the brain. So that's what we call the different gene expression at tissue level. We call it a tissue-specific gene expression. Then can you imagine the mechanism which enables this tissue-specific gene expression? I gave you already the hint, clue is by playing around the different transcription factors. And transcription factors are very specific to different genes. So in other words, you make very specific transcription factors only available in liver, whereas in your brain, another different type of transcription factors are only available in liver, and accordingly, those related genes are specifically activated in those related different tissues, is what this tissue-specific gene expression is going on. And developmental specific gene expression is, on the other hand, those differences are being made according to the different time along your growth span, or in other words, which is development. And even environment-specific gene induction is certain genes largely inactive, remain inactive, but upon very specific environmental cue, this gene is only activated type of this. A very good example is Hisha protein. As this name suggests, these Hisha proteins are only made when cells are exposed to highly elevated temperature. And certain mysterious proteins are just made out of such condition. That's why it earned its name Hisha proteins. It's everywhere. Bacteria, plant, fungi, animal, they all have these Hisha proteins. And universally, they have the same characteristic of this heat, high temperature, inducibility. How? By modulating certain specific heat-responsive specific transcription factors, these genes expression can be controlled. Then you can achieve this. How many of you? How many of you, without having any problem, follow what I have just said today's lecture about this? To me, there's nothing. There's no complexity or difficulty involved, even though this is not easy, straightforward concept, but actually, based upon your common sense, there's no reason for you to follow in understanding this, but that's just me. Your perceptive side could have a different idea or opinion. So that's why I'm curious, asking. Is it too much? Is it so difficult for you to understand until now? Then just tell me. Let me know. Let me know by writing down, hey, that's too much. Then I will try to reroute this. But to me, so far, I think try to explain only very basic terminology about this thing. On the other hand, without exposing these things, actually, there is no other way around. I can actually explain the actual what's going on in terms of so-called this gene expression, differential gene expression. Then how are we going to be able to understand actual what's the gene expression and how they are manage it? So that's why I'm touching on this. So this is actually a very famous figure regarding this tissue-specific gene expression appearing on the general biology textbook. So here are two examples of a liver cell versus a lens cell. Lens cell is in an eye cell, there's a lens. So in lens, in order to make up a lens, you need a very specific protein called the crystalline protein. And in the liver, another such the symbolic hallmark protein of liver is albumin. Albumin protein is a very famous protein and is very abundant in your blood. But actually, the place of synthesis is the liver and then dumped into the bloodstream. So this albumin protein is actually the regular resident protein in your blood. So anyway, so if you consider this, the difference between albumin protein and crystalline protein, whether even you look at the lens cell, they do have these both genes, albumin as well as crystalline gene. Just like hemoglobin gene, although this hemoglobin protein is made only in your red blood cell, but it's everywhere. Whether you look at in the skin cell or brain cell, that exact same hemoglobin gene exists, it's there, but simply doing nothing. They are inactive. Only this hemoglobin gene is active only in your red blood cell. That's why you can have this product, which is the hemoglobin protein in your red blood cell. And just like that, same thing. In the liver, although when you compare this lens and liver, both cells possess this albumin as well as the crystalline gene, but only crystalline the gene is active in lens cell, whereas albumin gene is only active in liver cell. How? This figure explains how. So in these two genes, in the case of liver, the crystalline gene is not expressed because it's empty. In this regulatory area is empty. There is no transcription factors available. On the other hand, the transcription factors needed for the albumin gene expression is plenty over there in the liver. And this actually figure also shows the secret of how some of those transcription factors that are distantly located, far upstream, can have an influence on this RNA polymerase by the figure is cut off in the left side, but by making this loop. This DNA can make a loop so that actual very intimate contact can be made from a long distance by looping, just like even though my finger is far away from this head area, but I can always just bend around this so that make this finger area touch this head area, and DNA. So can this DNA do this. That's how such transcription factors located far, far away from the actual transcription start site can also make this control the effect. So anyway, this is the case in the liver. Only the albumin gene transcription factors are available in terms of activity. Exactly opposite thing is happening in the lens cell. In the lens cell, only crystalline gene specific transcription factors are available, so they are very busy, crowded. But the albumin gene area, they are simply empty. Nobody is there. So you cannot have any gene expression because of that. So that's the differences. So whether you can have a specific transcription factors, whether it's an activity or actual availability, is the one that make the difference of this. So that's about the regulatory area of the gene expression. So this splicing of eukaryotic messenger RNA is something that the transcribed area story. So I will just put this thing off till Wednesday. And today, I will, for the rest of about 20 minutes left, let's take a look at this, the question. Question. There's no special question at the moment, but simply I like these two drawings. For this one, I'm just curious why this lady is so sad about, because this is poor mice that killed. And another thing is, I bragged about my drawings, and that humbled me. Okay, sorry, I will not brag any my drawing anymore. Oh, look at this. This instant drawing. You have obviously literary talent in this. So, okay. So this thing, how do bacteria select a specific host cell to infect? Specificity. This all involves in the whether it's a bacteriophage or not it's a bacteriophage. It's a bacterial thing. For unlike a virus has a very host-specific things through this membrane protein in recognition, interaction, specific membrane protein virus can recognize and then grab and then try to snake in. But bacteria is huge, relatively huge, so it cannot exert such a specificity of a host. And another thing is, don't get confused, is viruses actually enter the host cell, but bacteria usually do not enter the host cell. They simply enter the host tissues, usually. Although some bacteria actually even enter inside of the specific cell, just like a tuberculosis. Okay? They enter inside of our white blood cell and make the troubles, but that is not the general characteristic. Most bacteria simply do not enter because they cannot, they usually don't. And outside they have such a thing. So the host specificity usually bacteria do not have. But some very special occasions when they have such host specificity, always it's just like the case of a virus. They also find this host specificity on some specific cell surface receptor like. So if the host cell has a very specific protein, recognizable protein, a moiety structure, then another bacterial cell surface protein can recognize same thing, specific interaction. And that somehow brought about this the special relationship. It's simply whether the vulnerability-wise, some very immune challenged or old cells, the reason why they are more vulnerable to this bacterial infection is simply because they have much lowered defense ability in terms of their immune response. That's the only thing, nothing special. So disease be diagnosed using bacteriophage that detect a specific bacteria? Good question, actually. How did you know that? There is something called a phage display. One very specific bacteriophage on particular bacteriophage surface. Remember, bacteriophage is usually outside made of a protein. Inside, they have a genetic material. So we can attach the specific outside of this bacteriophage, some of very specific protein of our own choice. Okay? It could be different antibodies or different some receptor proteins, you name it. And then you test. Okay? So this specific, whether it's an antibody or any receptor, then you can utilize this thing as some diagnostic tools to, because they are, both of them are something that provide this specific interaction, the grabbing, binding interaction. That's how this phage display works. And actually the selecting the proper corona vaccine was also like this phage display technique was also involved in screening which type of vaccines are actually more suitable in such different types of things. This has been employed. This A, C, this complementary base pairing, I just went over quickly. Okay, there is a complementary base pairing specific relationship exists to some of the majors. Okay, it seems to be so obvious, natural, but to some of those non-majors, yeah, questions, why can't they? It's just like this. You bring your electric plug to the US, it doesn't fit because the plug hole number is different. Here we have 3 plugs, but if you go to US, there is only 2 plugs. Oh, the other way around. This is Korea and this is US. Anyway, it doesn't fit. Just like that, here, guanine and cytosine always pairs, whereas A and T, adenine, thymine always pairs. The reason is here, 1, 2, 3, these all indicate hydrogen bond. Between these two, they can have 3 hydrogen bonds structurally perfectly fit in their dimension. So if you're trying to bring this adenine, instead of this C, try to fit in guanine to force this complementary base pairing, and energetically it's so unfavorable. It doesn't fit. So usually you can't. So it's just we usually explain this by simply saying, okay, between G and C, there are 3 hydrogen bonds are being made usually, and between A and T, only 2 hydrogen bonds are made. That's why A and T always pairs and the G and C always pairs. Yeah, it is correct explanation. But if you actually further, in more detail, try to explain, because of those structural constraints. Okay? So if you try to align, flip around and try to align this A to in this, and it doesn't fit. If this complementary basing rule is violated, then exactly that's a mutation. It will create the mutation in the next round, because this complementary base pairing thing is so important during the DNA replication. Okay. A lot of times, actually, it can they have temporarily the wrong base pair can be made. Although structurally they are unfavorable, but a lot of different things happen in the cell. And if this structurally weird this thing during the replication simply go unnoticed, then mutation. The next round, then automatically, at least one of these DNA synthesis will become different DNA nucleotide during this replication. And the change, what's the definition of a mutation? Change in the DNA sequence. You simply accidentally changed this DNA sequence arrangement. Why use this uracil instead of thymine? Actually, the question should be made the other way around. Why use thymine instead of uracil in DNA? Why? Because who came first? RNA came first. Remember RNA world hypothesis? RNA was the original genetic material. And then gradually was replaced by DNA because of the intrinsic RNA problem. RNA was chemically very unstable. So in the beginning, RNA was the only possible material to serve as a genetic material because it can also provide at the same time the enzymatic function, ribozyme. But when you don't need any such enzymatic function at the same time, then RNA was discarded, dumped. Okay? RNA was betrayed because DNA was able to provide a much more stable, reliable function as a genetic material. So actually question should be addressed like why, when now the DNA became a genetic material, instead of using uracil, why did you dump uracil and then decide to use thymine instead? That should have been the correct question. Uracil was there originally. Actually, original combination, the sets of 4 nucleotide in RNA was AGCU. But then DNA became the genetic material, and then somehow, hey, uracil, you're out, you're fired, and why don't you come over, thymine, and the new member joined in these 4 nucleotide sets. Why? Then now we ask why? Why? What kind of disadvantage was there? Instead of, if we keep using uracil, what would have been the problem? Yeah, there was obviously a problem was there because remember, cytosine spontaneously through this so-called deamination reaction. Watson and Crick recognized this problem, spontaneous mutation of DNA. So cytosine can be spontaneously, without doing anything, can become a uracil. Yeah, cytosine can change into uracil spontaneously. Now what? You have a problem. Because if this cytosine changed to uracil, now originally this C and G was pairing with, and then this cytosine changed the uracil and G, UG pair was there, and DNA synthesis enzyme patrolling this and U and G, what's going on? Who is the correct one? And both of them will argue, hey, I am the original. Because DNA polymerase should take out, because DNA polymerase has an ability of a proofreading function. Incorrect base pairing, they removed and then correct this mistake, just like our typo spell checking problem, the function. But in this case, you have a problem over this. Which one is actually the correct one? Once they are made, whether this G was the original or U was the original, if the uracil was still one of our 4 nucleotides. Now, luckily, instead of uracil, we have a thymine. So now you have some kind of clue. Whenever you see this U, hey, why are you here? This is not a place with you are around. So we can easily, safely get rid of this uracil because uracil has no place in the DNA. So by employing this changed nucleotide, we are the cell could significantly reduce the mutation rate. Whenever this tyrosine conversion happen, tyrosine automatically become uracil can happen. But no problem, because we can get rid of this uracil. Oh, that's the problem. That's the wrong base pairs. Although, even with that, we have still some significant level of those mutation rate. But if this was also around, then the rate of mutation would have been much higher. How did we figure out the correct RNA secondary structure? It is actually because all this pairing is based upon what? Complementary base pairing. At least you know at least which nucleotide can have a potentially pair with which nucleotide. Now the second question is calculation, energy. So it's thermodynamically the least amount of energy, the free energy change. So you don't know about the free energy, whatever. It's there actually in our lecture, in the previous lectures, it's there. Anyway, there is some kind of physical the clue rule that you can calculate which is the most stable type of chemical bond by simple just thermodynamic energy calculation. So you can predict which area, which positions RNA can have this base pairing. And in these modern days, even the most sophisticated program, including the AI, can correctly predict this RNA secondary structure too. And actually the AlphaFold, the three-dimensional structure prediction tool is actually also coming from that basis. You have to calculate when you have which part of this connection, the chemical connection, would provide the least amount of energy. Okay? So more stable, in other words, more stable structure will be generated. And actually AlphaFold calculators. Although those calculations, because of so many different the cases in highly sophisticated advanced computers will lack the enough computing power to accomplish such things. So up until now, that has been largely impossible. But in these days, the huge, powerful artificial intelligence can provide such a huge computing power that has been available. So basically, that's it. This one is something. Messenger RNA is hiding to the microRNA. Is anything? No, it is not something that is being hidden by. It's just also another complementary base pairing. So probably the students is the question is coming out of this figures. There is microRNA and there is a messenger RNA. So hiding this particular sequence can have a base pair, complementary base pair with this particular area through this area single-strand RNA and this messenger RNA single-strand RNA, so they can have this hybrid RNA double-strand hybrid. And once this thing was made, and the RISC, this is a protein. RISC is a protein. So this protein is basically the cutting enzyme. So if this microRNA pairs with this specific messenger RNA sequence in this region, and this RISC complex, this protein, can chew up this RNA so that this gene, messenger RNA, become non-functional. Is the job the RISC complex and microRNA is doing. So it's not covering up, so hidden. So is this something to do with the recessive gene? No, it is nothing to do with such recessiveness thing or dominant thing. It's simply by binding through complementary base pairing and inhibiting this function is what this microRNA's job. So actually this RISC complex, how is it generated? It's a complex story, which is not the actual focus, should be the subject of the discourse. But anyway, to give you a snapshot of how it's being made. Initially, microRNA is produced in this kind of a looped double-stranded RNA form, and through some kind of processing, it became finally a single-stranded form. And at a given point, this single-stranded RNA can pair with the target, actual cellular target sequence, messenger RNA target sequence, because they are complementary to in their sequence. And in the meantime, these proteins are coming somewhere different place, and then they grab this complex. And what did I tell you about this RISC complex? They are the cleavage enzyme. So any those double-stranded RNA region is specifically digested, cleaved by this RISC. This is a huge protein complex. Different proteins are actually made up this so-called whatever RISC protein complex. So that's how. So you have this microRNA is one set, and another protein sector called the RISC complex get together and function. This. And quickly, the answer is, depending on actually how perfect this base pairing is. If this microRNA sequence and the target messenger RNA sequence is 100% identical, like complementary, then usually they go through this digestion route. And if they have some of this mispaired, unpaired area, then usually they go this inhibition of translation route for some reason. Okay, simply to answer the question is just like that. So time's up. So see you on Wednesday.